Again. All right. Good afternoon.
Okay.
And welcome to the fourth or fifth meeting of this series on interfaces between theory and experiment in brain sciences, which is supposed to bring together two distinguished scientists who work on related similar problems from both sides of the story. And it's really a special honor and for us to have today both Haim and Haim Sompolinsky, whom we all know, and Bob Shapley, who is coming to visit us. Actually, we're going to make him work hard for this visit. So starting with this exchange or duet or dialogue with Haim on what experimentalists can ask theorists and what theorists can ask experimentalists, in particular I guess around the questions that Bob is interested in, which is primary vision in general. Later on today at 5 we have the ICNC seminar, which is moved from Thursday to Sunday because some of us will not be here during the rest of the week. But before, so this is going to be another event where Bob is going to tell us more about his work. And as you all know, he's also our invited Hella lecturer this week, and will give us a more public-oriented, I suppose, talk on Tuesday evening as a Hella lecture. So I'm sure we'll all know something about Bob's work by the end of the week. And without further ado, I just invite Bob to tell us, to give us the first part of this dialogue. Let's put the microphone on as well.
So I have to tell you that the humorous aspects of what Tali was talking about have not escaped me. That I'm going to be sort of talking nonstop to you for days. And just as a preview, this evening I'm going to be talking to you about some work on receptive field analysis of neurons in the primary visual cortex, with the motivation that we have a kind of revolutionary concept of what's going on in the primary cortex, and I'll show you the evidence why we have this idea. And then on Tuesday, the plan of that talk is to talk about color and how color is represented in the visual system, and in fact how color perception depends upon computations that are going on also in early cortex. And in a sense we're filling in the gap of what are the mechanisms for a lot of the primary functions of color vision. And the truth is that there is a connection between the two lectures, but it's not obvious, so I hope we'll make it obvious as we go on. Now, after talking about those two things, there wasn't a lot left but Haim and I did consult about how we could structure today's dialogue, and the idea was to try to discuss future directions. So where do we think the field is going, or where do we think it needs to go, and what are open questions and what are mysteries that are needed to understand. And of course we'll draw on our experience and our expertise to explain how we think it should be attacked. But in fact, it's the nature of the discussion that we're going to be talking about topics that we don't know the answers to. So the thing tonight and the thing on Tuesday, the Heller lecture on Tuesday night, I actually have answers that I can give you about certain, I can be very specific about questions and answers, and structure it in a way that's very expected. It's not, the answers aren't expected, but the structure is expected, so it won't be unfamiliar. But here, we're talking about a situation where, we're really predicting the future, but we can't, of course. If we could, we make a lot of money. But, and so what we're trying to do is to, excuse me, what we're trying to do is to look into the future. And so what I'm going to do is talk about how I think the future will go, and based on where we are now. Could be wrong, but this is my best guess. And we want to see where what are the big questions that need to be answered, and what do we think are the kind of ways that the answers are going to be framed. And so my view of the big question is, how can the cerebral cortex act to do pattern recognition? What does it mean for it to do pattern recognition, and what's its style of computation to do pattern recognition? And then particularly, I'm going to emphasize that I think one of the big features of cortical computation that's going to be important in the future to understand is how feedback from different stages of processing to early cortex plays an important role in recognition. And so there's already lots of evidence about feedback, but I'm going to try to show you an example of where I think the feedback plays a big role. So I'm going to choose an example that I think is particularly salient, and then I'm going to try to generalize to say, okay, we have a pretty good understanding of how this works in the visual system under these particular conditions. I'm going to try to generalize to everything. You could question that, and that's a perfectly legitimate question, but that's my plan, anyway. So what I want to talk about, is the issue of what the brain has to face when it's doing pattern recognition. And this is a very familiar picture to many of you, I'm sure, the face vase picture from Edgar Rubin. And it illustrates, that's good. Anyway, it illustrates ambiguity. Well, I guess that illustrates ambiguity right there. That there are many possible solutions to pictures that you look at. So of course you know that you can see this as a light blue vase sitting in front of a dark purplish background, or you can see it as two dark purple faces in profile looking at each other. Two prime ministers or somebody, and if you look at it for a while, if you keep on looking at it, you see the two faces or the vase come back and forth. It sort of goes back and forth. You get these reversals, so-called figure ground reversals. That's a technical term for just the experience that you experience. And what this is illustrating is that pictures like this one, and this is not there are millions of these, but this is the famous one. Pictures like these are totally ambiguous. That is to say, they admit two perfectly good explanations. There may be more than two, but two big ones that everybody sees automatically. And this is what the brain is facing all the time. It's just that we're drawing it out and making a big deal of it here. But in general, things are ambiguous. And what has been observed is that the brain always goes and finds an answer. That is, it never says at any one moment in time there are two things. It always says it's the faces or it's the vase, and it goes back and forth. So I'm not focusing so much on figure ground here as on the fact that the brain is facing ambiguity, and it always solves it moment by moment, although the solution may not be stable. So here it's not stable. It keeps on breaking. But so this this issue comes up, and then the question is, what are the ways that the brain deals with ambiguity, and it's not only how it deals with ambiguity, but also how does it find an answer? That is, why is it always finding one answer and not two, or if there are three possibles, three, or so on? It's always going for a single answer. This phenomenon of always finding an answer in the case of ambiguous situations is also very nicely illustrated in a completely different context in the work of Hans Wallach, who, I don't know if any of you have heard of his name. He was a famous 20th century psychologist, perceptual psychologist, who studied motion he studied the motion of grading patterns in apertures. So you would have patterns like this, and they'd be moving. Well, you would see them moving like this, or you could see them moving like that. But in any case, when you're looking at a moving pattern in an aperture, the actual direction of motion is continuously ambiguous. That is to say, you could see the motion if the normal to the line is, as I've drawn it like this. The perceived direction could be anywhere in this half plane. Sort of, it's possible to physically. And but the thing is, at any one moment in time, you always see one direction of motion. And he reported on this as the most surprising finding of his work, which was that the brain always found a definite answer. The answer could change from time to time. So in fact, sometimes you'd see the motion going this way, sometimes you'd see the motion going this way, sometimes you could see the motion going that way. That is, the motion direction could vary, but at any one moment in time, there was only one motion direction. And so similarly, there's only one answer in the face vase problem. And in general, the question is, how does the brain get to find an answer? So that's, I think, one big issue. It always, so that, to make an analogy, the brain is always like a professor. Even if it doesn't know the answer, it comes up with an answer. Okay? So, and the question is, why are our brains all like professors? That's another way of framing the problem. So I wanted to talk about an illustrative problem in visual perception and visual neuroscience that can be used to study these questions of ambiguity and how the brain solves ambiguity and how that can guide us forward to understanding how pattern recognition can be done in general. So I'm going to talk about the visual perception of illusory contours here, because that's something I know about. And I can tell you a nice story about it. And I think it's actually very illustrative. It shows how the brain can deal with ambiguity and the interplay between very local, recurrent, and feedback signals. And also, it illustrates how you go from local to global processing, which is part of the issue of how I think we should think of how the brain works, is by combining lots of local signals by some rules for combination. So let's see. So here's a question of illusory contours, and this is an this slide is mostly to illustrate what the situation is, which is you have this sort of funny occluding shape in front of these black inducing figures, and it looks as though it's this picture here of a figure standing in front or occluding a bunch of circles, even though in fact the situation is ambiguous, because in fact it could also be just 5 odd-shaped things that happen to be lying on the table in a certain arrangement. But that's not how you often see it. You often see it as this sort of occluding object, and it's a taxing stimulus in terms of segmentation, but it's also a perfectly good example of ambiguity in perception. Now here, I think this is where it is. Yeah, I wanted to show this to you because this is an example of the same sort of pattern, and I wonder if anybody knows where it's from. Have you seen this? Most people have been where this pattern is located, in this room, I think, and you probably have seen it multiple times, but I don't know if you've noticed it. You see that there's this nice illusory contour here. There's a nice illusory contour here on the outside, and this disk, together with the illusory contour on the outside, give you this strong sense of an annulus standing in front of the objects, right? But I don't know if this is actually my drawing. I just did this on the computer, but it's my rendition of a pattern that's very familiar, or is at a place that's very familiar to many of you. Anybody know? Anybody recognize it all? Want to guess?
Like the shadows of the Washington Monuments?
No, actually, that's a good one. But it's you could see so these sometimes you see things in shadows like these. This is actually my very crude rendition, it's not very artistic, of one of the tiles on the floor of Beit Bialik in Tel Aviv. They have these very elaborate Italian tilework floors. And the Italians are very artistic, and they make these patterns, graphic patterns. And so they generate these very strong illusory contours. I just thought you might be amused that you can find it close to home. These patterns are found everywhere. They're found in nature, of course, also. You see such things happening. It's a kind of thing that you wonder a little bit why the brain is designed to solve this problem, but in any case, it's something that comes up in conditions of low vision or fog or other cases where contours are degraded, and you kind of fill them in. So the actual, there's no, just to explain for people who may wonder what I'm talking about, there's no physical difference across this boundary or this boundary or this boundary, but it looks as though there's a clear boundary connecting all those regions, especially if you're sitting more to the back. Those in front are might be a little harder. So anyway, these illusory occluding type surfaces are present often. In this case, actually, it's an interesting case of ambiguity because one interpretation is that this annulus is sort of like standing in front of the whole pattern, and another interpretation is that this disk is in front, and then this thing, and then the other ones are behind. So there's like three levels, and you can get multiple interpretations. This is again another example of ambiguity in visual patterns. So the question is, how does the brain do this? What's going on, and what are the mechanisms that accomplish this function? And a long time ago, I got involved in some experiments on the psychophysical side of this with a student named Dario Ringach. Many of you may know him. I don't know if you've heard of him. And actually, this was a pre-PhD project that he worked on. He was an amazing guy. He's an amazing guy. And these other people all worked on this notion of how do you measure over what distance you can get completion, say from this side to this side, or from this region here to this region here. How long is the completion? Over what gap can completion occur? And actually, you can measure that it can occur over as much as 10 degrees of visual angle, which is a big block for those of you in the front. It's about this big, and in the back, it's about this big. It's a big region of visual space. So the question is, how can you do that? Because you can get, when you finally measure it, you find out you can complete over a very long distance. And so it's possible that segmentation in this illusory contour context relies on cells with large receptive fields. And that's certainly one possibility that people have proposed, is that there are elements that have very big receptive fields that can knit together signals from very long distances, and you can sort of see the completed figure by summing up the signals from all over the place. But there's evidence for small receptive fields because there was already neurophysiological work from a long time ago, more than 25 years ago, about how early visual cortex can be involved in responding to these patterns. And this may be, some of you may have delved into the history of this or know about that van der Heijden and Peter Hans in the late 80s and even before this did experiments with single cells in monkeys where this little ellipse is supposedly the receptive field of the neuron in the monkey, and if you have a figure that covers the whole receptive field and moves across it, you get a response. The spike responses, as indicated by the little white dots on the black background, look like this. These are, individual raster traces. And then, if you have a gap between the two ends of the moving bar, and there's like a blank region over the receptive field, you can still get an evoked response. But if the gap is too large, or if there's an offset between them, then there are no spikes evoked. So there's a kind of completion phenomenon you can see in individual neuron receptive fields in V2. And there's also some evidence you can see these things also in V1. Not this kind of, but other kinds of illusory contours. And then also, another piece of evidence is that this whole phenomenon of seeing illusory surfaces is very sensitive to small changes in image properties that could only be sensed at very high resolution. So this is some work from a man named Mnguzzi, an Italian psychologist, who studied these sorts of patterns where there's a nice occluding disk in front of the bars. And then if you just round off the corners, so you just smudge the corners a little bit here, just a little bit, you get this figure where you hardly see any occluding figure at all. It's like it's just open space. And so very small changes in details of the image can have a huge effect on the qualitative phenomenon. So that's another observation that suggests that there could be early cortex could be involved in this. And so there's two observations. One is that there's very wide gaps across which you can fill, and the other observation is that you also depend upon very high spatial resolution information, which is usually only available in early stages. So there's a puzzle of how do you do both things? How do you simultaneously satisfy both requirements? It seems like it's impossible, or it's some kind of a trick. There are all these tricks that the brain has. So there is a proposed solution, which is that, in fact, there are small receptor fields. They're involved, but they're involved like in a daisy chain or a cascade, where they interact with each other along a distance, but that this interaction is guided by feedback. So this is where the feedback is coming in. This is where I'm finally getting to my main theme. I'm sort of divulging where I'm going, is that feedback from higher cortical areas guide this filling in. So the filling in is done at an early stage where local information can make a difference, but the overall trajectory is guided from some higher level where signals, so that you can have your cake and eat it too. That is to say, you have high resolution, but you also have global completion by having feedback guide the high resolution information. So the cells in V1 and V2 give you the direct high resolution information, and they also give you a substrate on which to represent all possible contours. So that the map of the spatial map in early cortex gives you the wherewithal to create cascades of any particular shape. So one thing I haven't said, but it actually is obvious from all the way back to here, is that you can have filling in with, it doesn't have to be straight. They can be curved. Interpolation of this kind, these filling in things can be curved. And like here also, you get curvature. It doesn't have to be totally straight. So you can smooth things so that here, when we say that you need a cascade which could represent all possible completed contours, it's all possible sort of smoothly curved ones. That you can represent smoothly curved changes, but not very rapid ones. But they don't have to be exactly straight. The requirement for straightness is not rigid. And that's because you have this sort of high-resolution map of the world onto the visual cortex, which allows you to have positions that are not just completely aligned along a straight line, to be combined with each other. Okay. So a lot of the way that these ideas have been tested is with work with humans using human fMRI experiments on illusory contours. And a lot of the action takes place in the early visual areas. And we were looking for early visual cortex involvement. So let's look at the fMRI evidence about it. This is an older review by Grill-Spector and Malik talking about the mappable visual areas. And the actual technical part of the slide is here. These are the mapping tools, which involves radial growth of patterns or rotating patterns, which allow you to create a retinotopic or visuotopic map. I call it visuotopic that is a map of the visual world onto the cortex. And so the bluish purple is sort of mapped. So there's a purplish thing in the middle here, which is mapped along the line of all of these various visual areas, and different visual angles are denoted with the different colors here. And the visual areas this is now one of these flat maps that fMRI people like to use, showing in a human the posterior cortex, visual area 1 at the most posterior part of the cortex, and then as you go more interior, the different V areas. This would be, the same picture originally, before it got unfolded, and then this is the folded version of this sort of map of the visual world onto the posterior cortex. And all of these areas, V1, V2, V3, VP, and so on, these are regions that have visuotopic maps, where the whole visual field is mapped in an orderly array, and particularly in V1 and V2, it's a highly detailed map which allows this cascade, as we proposed it, to be filled in on. And then more anterior, this is again one of these folded maps, flattened maps, where V1 and V2 are more in the posterior part of the cortex, so we're going anterior. And as you go anterior to the visuotopically mapped areas, you get these areas like LO and HV4 and P fusiform, the fusiform gyrus, where in fact you have activity which is connected with structural order. So this region of yellow on both sides is the region of fMRI activation where a structure, indicated as a modern art sculpture, is compared to random noise. So those are the two conditions that are compared. And you subtract the activation caused by modern art from the activation caused by noise, you get the yellow areas, which are these sort of more anterior, non-visuotopically mapped areas. And the reason I point this out is that this area, this LO area, is in fact a higher visual area that seems to be involved in the resolution of the ambiguity of the illusory contour perception. So the fact that it's defined not in terms of illusory contours at all, but in terms of the fact that it likes patterns or structure more than it likes pure random contrast. But in fact, when you run an illusory contour stimulus, and that the illusory contour isn't shown, but it's something like the illusory shapes that I've shown you before on human subjects, you get activation. So this is now not a flattened view. This is a slice through the brain like this, a coronal view. And this white outline, which you can see best, I guess, here, this white outlined area corresponds to the LO region. So I'm just telling you, the definition of the white outlined area in the next slide is the LO region from this slide, the area that's sort of structural. And this area here is activated by illusory contours, so illusory shapes like the ones that I've shown you before. All the examples that I showed you would generate activation in these more anterior, higher level, mid-level visual areas that have large receptive fields and are sensitive to structure. The thing is that when you do this experiment with illusory contours, you get activation in LO. When you do the experiment with rounded corners, the ones that were used in that study weren't this particular one, but it's the same idea. You round the corners in the same way that Menguzzi did it. You get the same activation, in fact. So, in fact, what had been thought for years, including from my own work, that the activation in LO here was due to illusory contours, in fact wasn't. It was, in fact, due to the activation of neurons that were sensitive to the idea that this region was interesting, or this was a salient region of interest. So, in fact, the higher order areas respond the same to this and to this. And what makes this percept different from this one, what gives it its flavor of a completed illusory surface, is in fact thought to be the interaction between the lateral occipital cortex responding to the salient region and the early visual area responding to the local features. The guided cascade of completion in the early area is guided by the downward, the top-down signals from LOC, and that's this is the thesis of this paper from 2003. What you need to prove this is experiments like this, which show that you get equivalent responses with both illusory surfaces and with salient regions, and you need experiments to show that you get activation in V1 under similar conditions. And that was supplied later by these people, Mertens and Pullman, who showed that, in fact, so that you have to take my word for it because you can't really see it so well, but this is actually the Calcarein Fisher, and this is V1. And they found that in subjects who learn to see illusory surfaces, you can teach people to do it, and only the ones who learned, that is, so here are the learners, and so there's an improvement from beginning to learning, and this is a non-learner. Only in the learners, you actually get this sort of activation of primary visual cortex by illusory surfaces. So all the conditions for this scheme are fulfilled. That is to say, you know that the LOC is activated by the salient region formed by the inducers, and you can show that when you have illusory surfaces, V1 is activated, and the conditions for this top-down processing are in place. So we can imagine, so I take this as a paradigm. This is like, this is one example that at least we know about of how top-down effects can actually guide some kind of pattern recognition problem where ambiguity is involved. And indeed, you can see that this kind of scenario solves the problem, the problem of fitting both constraints. You need the LOC component to allow you to fill in over large areas and to have to enable to complete over large gaps. And you need the V1, V2 component in order to be able to respond to fine detail and to high image resolution. So that it's a very nice paradigm for how it might be that you can go from local to global and back again in ordinary pattern recognition, and also how ambiguous patterns could be reconciled by combining signals across different scales. So there are more reasons than I've shown to believe that V1 is involved in illusory contours. There are direct experiments with different kinds of patterns from our lab and from Tai-Sing Lee's lab that show that V1 can be activated under conditions where you see illusory surfaces. And there's human psychophysics that shows in various ways that, for instance, the perceptual learning that Nava Rubin and Ken Nakayama and I studied of learning to see illusory contours can happen very rapidly. And the explanation that we came up with for that kind of very rapid perceptual learning is that there's a combination of both bottom-up signaling by local signaling mechanisms, but also top-down influences from LOC and other areas where remembered shapes can speed the acquisition of perceptual learning. There's also an experiment that Pillow and Rubin did later where they showed that there was a cost if you had to complete an illusory contour across the vertical meridian. It took more time if you had to, because you have to combine signals across the two visuotopically mapped hemispheres. It takes more time to complete a contour this way than to complete a contour this way, which is all within one hemisphere. And there's a third study that shows by Mertens and Pullman, a later study by the same group in Germany, that there's an extra cost for filling in an illusory contour through the blind spot. In fact, it's almost impossible when in a region where there are no neural local cascades in the cortex, where there's no local representation. So it isn't simply that you can use large receptive fields that span across space. You need to actually go through the local representation. So there's lots of reasons to believe that V1 is involved in the illusory contours. And so, but because of the constraints of long-distance communication and large scale, there's also this idea that you couldn't do it just with V1 alone because that doesn't have the information about the large scale. You need the feedback. So the two constraints are actually very powerful constraints on how you can imagine this thing being implemented. Now, this is now a segue into what we could use this paradigm for as an example for in understanding pattern recognition more globally. And we're going to go back from human to monkey. So this is a monkey review, a review of monkey cortex with V1, V2, and TEO and TE. These are monkey terms for the infratemporal cortex, the sort of ventral pathway, the ventral stream of the monkey cortex. And the notion is that as you go more higher, it's even drawn lower to higher, in the ventral stream, that you get more complex visual features, and you get larger receptive fields, and you get slower responses. This is not really the whole story, but it's certainly true as far as it goes. But it's also true that the responses as you go. So this, just to remind you, these areas in the ventral stream of the monkey are sort of corresponding to these kind of areas in the human, sort of going from V1 up to LO, is sort of like going from V1 to through V4 to TEO in the monkey. And you find neurons in these infratemporal cortical regions that respond to patterns, various complex two-dimensional patterns, sometimes natural images versus other things. And one thing that's striking about them also is that not only is the response time slower, so there's a slowing down of the response in terms of its rise time. So latency refers to rise time, how slow it is after you present the stimulus, but also there is a greater amount of persistence. So it's as if the response lasts longer. So when you turn the stimulus off, it's not shown here, but when you turn the stimulus off, the persistence is much greater for these higher level areas. And I take that to mean that, in fact, we should really consider these different stages as not one set of filters after another becoming more elaborate filtering operations, but in fact that there's a combination, especially also just anatomically. If you think of where this is in the brain and where this is in the brain, you're moving from the posterior cortex to a region in the brain that's very closely associated with the temporal cortex, which is, of course, where we think memory is largely involved. And so the gradient here is sort of from the outside world to our memory, where I would say, the gradient of present input is highest here, and the contribution of memory is highest here, and there's sort of like a continuum of improving or increasing amounts of memory contribution to the response versus increasing amounts of, down here, of present environment input to the response. And so what I'd like to propose is that this paradigm that we've been discussing about illusory contours and completion and so on may be, in general, what's used in any kind of pattern recognition situation where top-down influences, which have a greater and greater component of memory, may be modulating what is initially a purely external input phenomenon. So that's one aspect. And the other aspect of the final solution, the complete solution, I can't say the final solution, the complete solution, is that another issue or another thing to be concerned about is what are these higher level areas like? So there are, I'm sure everybody knows about the face area and the place area, and the house area and the chair area, and so on. And so here's a study from Ishai et al. From 1998 showing houses and chairs and faces, right? Yeah, and just this is just a replication on the other side of the brain. And what you see is that in different areas, so in the medial fusiform gyrus, in the lateral fusiform gyrus, in the inferior temporal gyrus, there are different. So this is again, in humans, you're seeing different human reactions to the different stimuli. And in the let me before I step out, I want to see the faces is red. So here's the face area, the lateral fusiform gyrus, and you see that its response to faces compared to the response to houses or to chairs looks like this. The two different traces, by the way, the lower trace is always passive viewing, and the upper trace is some kind of task, just to remind you. And you see that, in fact, in the most selective face area, in fact, the response to faces might be twice as big as the response to a house or to a chair. So you could say, at least some chairs might remind you of someone's face, or vice versa. And similarly in the house area, you could say, well, it's true that it responds mostly to houses, but you could say, well, that face reminds me of a house, or it has some similarity to the house that I have. And you could look at this and say, oh, it has some face-like properties. And, but the main point of the paper, and I'm just echoing their argument, is that the specificity is not absolute, but relative. And in fact, there's fairly broad specificity of each of these areas, and presumably the neurons in the areas. And so there may be, there may be highly specific occasions, but in a lot of places there'll be broad general specificity. And so I think that one issue, which I think I hope that Haim will address, is the issue of how this kind of result, and of course there's lots of other results like this throughout all the visual literature. I just chose this because it's sort of so salient to pattern recognition, is that these this leads to the notion that you need population coding, that you can't do everything just with a single neurons, that it isn't like one neuron rings a bell and says, oh, that's Johnny, or that's Haim, but rather that there's some kind of bell-shaped distribution on some energies, on some surface of activity, which then needs to be identified with the object of interest. Right, and this is again going back to these regions. Here, in this fusiform gyrus is just the same kind of region that we're discussing here. So this is just I'm just linking the pictures. So I'm linking this picture with this picture, just so you recognize that although this is a coronal view and this is this sort of flattened view, it's the same area that's involved. And the question is, what do these LOC or fusiform areas, what is their function? What are they doing? And this is where I'm going to now go off the rails and depart from my script and just say, it's in my script, but I'm going to depart from my from any sort of firm support and go out way on a limb and make a modest proposal, which is that this is for discussion purposes only. I'm not like I'm
supposed to challenge the theory.
That's right. So this is my challenge, okay, as it were. It's not really a challenge. It's sort of like I'm going to make a statement and you can attack it if you want. That's fine with me. And that is that I'm going to suggest that higher visual areas are memories for templates. That in fact, it isn't true that they're really, strictly speaking, visual filter areas at all. And I'm going to suggest that perception in general is a kind of associative memory, where you're trying to actually not just simply respond to anything, but in fact you only respond to the things that you know. It's an old idea in philosophy, that you can only see the things that you know already. But I'm going to make it specific by saying the reason why you only see the things you know already is because those are the only things you possibly could see, because that's what perception is the association of what you know already with what's coming in. And what I'm suggesting is V1 is the current environment, always, that is to say, it's providing the present outside world, and the higher visual areas are the stored images. And in order to link the present with the past, which is what I say perception is, linking the present with the past is what I'm saying perception is, that you need feedback. That is, it can't be done without some kind of top-down talking. So bottom-up and top-down signals must be involved in some kind of loop structure in order to, according to my way of thinking, according to my modest proposal. And therefore, feedback is necessary to link the stored with the current. And then, when you have this linkages and activation of different memories in this associated memory, then I suppose there needs to be some kind of winner-take-all mechanism. Now, the reason why for the winner-take-all, I'm going to go all the way back to what I said at the beginning, which is that in perception, you always come up with an answer. It's never true that there are two answers. There are two possible answers always, or there could be five, but you always come up with one answer at a time, and then they fight with each other back and forth. And it's some kind of winner-take-all or some kind of other interaction among the neurons that are representing the memories that I propose in my modest proposal are involved in perception. And then finally, broadly, two neurons in these higher visual areas require that there needs to be some kind of population interactions. It can't be done just neuron by neuron, but somehow, or there must be some kind of conduit for a population's activity to interact with another population's activity to allow for this competition between percepts, which are really competition between memories, which are somehow favored more by one input, by the present input. Whichever input is more favored by the present input wins the battle and becomes, at present, the winner, what you see. And then later it may get weaker and be overtaken by the other memory because of the present input, and it becomes then the one that you're seeing for the moment. So there are more questions. And I'm almost finished because I don't usually do this. There are questions which we haven't addressed at all, but need to be addressed at some point, which are, what are the functions of the different cortical laminae? Now, I've done a lot of work, and I'm going to be talking about it quite a bit tonight and a little bit on Tuesday, about, and I have to talk longer. I'm just kidding. I know. Anyway. On Tuesday, I'll also be talking somewhat about this issue, which is that you shouldn't really think of the cortex as a monolithic thing. It actually is multiple. It's sort of like a parallel processor where there are multiple functions overlaid on each other in different layers. And the question of how the different cortical laminae participate in this sort of bottom-up, top-down handshaking operation, I think will be something specifically that the experimentalists should contribute to in the future. Another thing is that I didn't really explicitly say it, but in this illusory contour problem, and in general in the ambiguous resolving ambiguity problem, surfaces and local features play a very large role. That is to say, what is actually remembered is often in terms of surfaces and features, not infinite amounts of detail. And so the question is how these are represented in populations of neurons in memory is, I think, an interesting question, and something. And so how you can get from local atomistic feature signals to signals that represent full surfaces is, I think, something that will be of theoretical interest. And I think that this previous question about features and surfaces may be related to why there are so many extrastriate visual areas, which is another puzzle. That is, why do you have so many of these areas? Why aren't there, one or two, or why isn't there one? A top-down area and a memory and a blackboard. And but it may be that the constraints of representing the perceptual organization of surfaces in space and features may be related to why there need to be so many extrastriate visual areas in humans and in monkeys. And the layer issue, I just wanted to point out that the layers are a big thing in V1, and I think it's generally ignored in other cortical areas just because nothing is known about them. But in V1, we know that the input layers have very different properties from the cortical output layers and from the layer 5 output layers that are going to the superior colliculus and the layer 6 output layers that are going to the geniculate, so that actually different functional properties of cells in different layers may be very important in the future. And that's it. So I'll just go back and say where I would end is sort of my contribution is a modest proposal, and then all these questions that come up as a consequence of it.
Thank you, Barbara. I just want to say that the idea that perception is about prediction, expectations, and it is not new in this forum. Anyway, a lot of us are talking about this type of model.
Good. That's great.
There's actually a whole course tomorrow morning about perception, action, cycle, which is about this particular filter. Anyway.
Good.
Maybe we take a 5-10 minute break and continue with Haim's answers those challenges to you.
Good.
And then discussion. Afterwards?
Well, it's your time. Yeah. So,
questions afterwards?
you want questions first? You want questions to both first?
I think we've been preempted. Let's go. let me remove. Where did he put it? I can't follow. I'm trying to unplug. Okay. I'm out of your way.
I'm sure that we need to meet and talk a little bit.
Okay. Well, you know where I am. Okay, go. Okay. Well, but at least I did fill out the time. I said I did fill out the time. Hey, nice to see you. Hi. Hey, that's great. I saw it. I was very pleased. I was very pleased you weren't asleep. Good. That's great. Oh, wait. I have to put this off. This is for you. This is for you.
נזכיר להם את הסיקוונס שלנו.
בסדר. רגע, אז אני מכניס אותך?
But that's called chemical engineering.
אוקיי, טוב, אוקיי, זה מה שלומך?
I don't know exactly. But on the other hand, the other hand is sewer and that's pretty cool. The other time I went to my younger daughter.
למה אתה לוקח את זה אם אתה לא יודע?
אני אומר, אולי תלמד, כי הרי אתה כל כך, אני מכיר אותך, אתה משקיע מעגלני כמה קצת הבנני פחות. אתה משקיע כל כך הרבה מאמץ על חצי שעה לתורמים שבכלל לא יודעים מה זה איניביציה. הם כאילו לא יודעים מה זה איניביציה.
לא, זה אני רציתי להגיד לך. יש לך חומר על איניביציה פופולרית קצת? כי אני חשבתי שחצי מדען זה בעיה.
לא יודעים מה בכלל הבן אדם מדבר.
אני חשבתי, אני כל כך חושב מה זה איניביציה.
הם חושבים שזה איניביציה שזה רחצן אותם שלא נשתנה.
אבל לא, אין לך משהו מה?
איזה רמה שזה? זה לא מילה.
אני יודע מה...
יש לי קצת על איניביציה. קצת איניביציה על חוט שדרה זה לא מילה.
לא, אני משתאים תיאורטיקאים, סוגים שונים של תאים,
אני בשקט שלי.
I'll take my water, and that's your water. Is that okay?
Yes, that's
I just need it for me and for also people.
ניגשת אליי כאילו, רוצה להגיד לי משהו.
עבד לי מעיל, אני חיפשתי אולי...
בדקת שם ב...
לא, חיפשתי שם.
Vision can deteriorate extremely fast, you know? I went to the doctor and he told me that they prescribed something else on cover. One and said that's mine. I went to an optician and they told me how much it cost. We got a sticker shot. And I was planning on getting one. And I was like, I get cheap glasses. How much do you think? What's your number? 1 and 45?
So just before we continue with Haim and the second side of this duel between theory and experiment, I just want to remind you that this is a series that started on last November and will end in June, and we are just in the middle of it. In principle, an ICNC course, and some of you actually are taking it for credit. The output of, or the requirement of this course is really the research proposal. Let's see how many can actually do that. So we have Yossi Haim on May, and then last end of May, Yadin Dudai and Misha Tzorich, and June 7th currently is Elon and Amir. And I'm still waiting from Udi and Yair to tell us if they want to do it at all. So, Haim, please. Working for free.
Okay.
Just put the microphone.
I hope the rector of the university did not hear your lecture, the beginning of the lecture, or your presentation, Bob, because she will question whether I deserve the title professor. Because unlike you, modular professor, I am going to confess already now that I do not understand neither what the brain is doing, but also not what V1 is doing. And I am going to, first of all, focus mostly on V1, although later on there will be inevitably stuff on other parts of the visual system. And secondly, since I'm not going to present a model or a theory about V1, what I thought, at least it was useful for me and maybe also for the students, I'm not sure about the researchers here, to take stock, to see what is out there in terms of models of V1. What are the approaches, old but more interestingly new approaches about understanding V1? What are the problems or limitations of each one of those approaches? And a little bit of where shall we go from here, or what is needed, or what are the outstanding questions, etc. So this is more or less my presentation. I'll start from the classical approach to V1, namely receptive field theories, which have been quite successful to explain many of the properties of responses in V1 in early vision in general. And I'm showing here several layers of such approaches. Again, I'm assuming most of you are familiar with this. I'm just giving most of my talk will be more like bullet points, because there's something to remind as reminders for you of things that you have learned in courses and seminars and so on. So starting from the retina, LGN, receptive fields, the more interesting, so to speak, receptive fields in visual cortex, simple cells in V1, oriented receptive fields, Gabor filters. Formally, from the point of view of mathematics or modeling, they fall into what's known as LN model, linear nonlinear model. You have a neuron that is described by a linear spatial temporal filter of the input of the image, and then an output nonlinearity, and to describe the actual firing firing rate or response of a neuron, a simple cell in V1 to an image. So that's sort of the classical description. Now there are more elaborate descriptions, mostly for complex cells. They are in this terminology that they will be L, then N, then L again, and then N. And let me give you an example here of what's known as an energy model for complex cell. You have now again a spatial temporal filter oriented filter of the signal of the image, maybe time dependent image. You have several filters. Each one of them undergoes nonlinearity. In the particular case of complex cells, this nonlinearity takes the form of a quadratic nonlinearity, so the cell responds to both polarities of the same filter. And then they are summed, and then again, maybe output spike nonlinearity. So that's a kind of more complex or elaborate combination of linear operations and nonlinear operations. Actually, more recently, there have been some interesting suggestions for generalizing this approach. rather to this particular kind of examples of a quadratic nonlinearity and two filters maybe, to a more general case where you describe the probability of spike given an image as some nonlinear function, general nonlinear function, of a set of filter outputs. Namely, you take the image S and you project it into filters W1, W2, up to WK. And basically the hypothesis is that the response of the neuron given an image is a function, is an arbitrary function of this subspace of the image given by those filters. So this is quite general, and anything you can model by it, except that the crucial assumption or the interest is if indeed the number k is small compared to the dimensionality of the stimulus. So this is sort of a general nonlinear function of a low-dimensional subspace of the image. So this will be sort of a more general formalization of this receptive field approach in general. Now, this kind of approach there's been a lot of interesting work how to compare such a model to data. And there are all kinds of reverse correlation, reverse covariance methods, and information theoretic methods which are related to it to find those informative filters. Okay, so this is basically the filter approach. Now, what is the goal of this model? What are we trying to achieve by this? So that's, I think, part of the difficulty in analyzing or surveying what has been done in modeling V1 is to try to understand what is actually the goal of the model, what that model is trying to do. Now, so I would say for the receptive field there are three dimensions, three axes, if you can think about it. I think the most immediate one is the phenomenology. In other words, to have some compact description of the response of single cells in V1. Simple, complex, but single cells in V1. And that's very important, because if you are not aiming at having some compact description, so it's not clear what you achieve by writing some model of the kind that I described. Now, there is also an implicit, sometimes explicit goal, is that actually to hint or maybe describe a mechanism of this receptive field. So most models of this approach really think about a feed-forward projection. So those receptive fields or filters are really coming from the patterns of feed-forward inputs to single neurons in V1, and the nonlinearities has to do with spike nonlinearities. Now, the more complicated models will involve more mechanisms, but so receptive fields, at least to some degree, and depends on who is doing what, and if you read the literature, but I think the mechanism underlying it is part of the part of the goal, and basically feed forward with some local nonlinearities. Now, there is another goal, or another axis which you think about in this function. And again, the receptive fields are implicit or explicit, has the picture, the concept is that this is what the neuron is doing. This is what the neuron is designed to do. This is why God actually created those neurons in this particular system, to report to downstream systems the output of this filter of the image. So it's a sort of a system of filter banks, which is describing what is the functionality, what is the role of this neuron in the visual system. The role is to report to downstream systems the output of this particular filter or set of filters in the current image. Okay, so that's simple. Now, what are the problems with that? There are several, numerous. Some of them Bob already mentioned. Some of them I think he will elaborate in his in your seminar today, actually, in fact. But so I'll be just brief and say that in general the problem is that when you come up with such a receptive field or filter model, you may do a very good job in explaining the response of the neuron to a restricted set of stimuli, ensemble of stimuli, but when you try to to test it on another set of stimuli, more complex set of stimuli, generally there are problems, failures, and the degree of failures depends on what is the kind of complex stimuli that you have. And the general picture that it looks like that. So this is the example of taking a receptive field, taking a neuron, this is an anesthetized cat, etc., measuring the best receptive field coming out of response to gratings. And here you see the spatiotemporal receptive field, kind of a Gabor receptive field with maybe some changes as a function of time, and then fit this, using this model, predict its response to a natural movie, to sequences of images mimicking eye movements in a scene. So it's rather reasonable model of a natural time-varying stimulus. And that's what you see. The red line, the solid line is the predicted response, and dashed line is the actual response. So you see you can quantify it by numbers, and the number is that the correlation coefficient is, I don't know, 36%. But you see the feel is that there's some qualitative discrepancy here, that the actual responses are sparser and stronger, appear sort of bursts in some specific episodes in the movie, in the sequence, and that's not what you see in the actual, in the predicted model. So that's an example and has been studied by many, those discrepancies. Now the rest is, in some sense, maybe excuses or origins of these discrepancies. So, for instance, it's clear that those models do not incorporate context. And as Bob already described, and again, he probably will elaborate more on his talks today or on Tuesday, that mostly that the question of context responses in V1, I think, is somewhat controversial. It's somewhat murky in my view, because if you look at, I think, at results coming from recordings from microelectrodes, spike responses in particular, the general impression is that in V1 you see rather modest effects of attention and context and so on. However, if you look at the functional MRI, then you see that for reasons which we can debate, we can argue about, the context influences in V1 are much more obvious. I don't know, again, why. Imagery is another example, extreme example, that V1 activity can be induced by imagery, again, primarily from functional MRI, and that's definitely something that is completely absent from this receptive field picture, and so on. The long range and feedback effects are completely missing. Temporal aspects of coding, usually it's a statics, or at least a fine rate, not more than that. And another is that it's sort of unsatisfactory in the sense that it doesn't tell us, it doesn't give us adequate, at least intellectual satisfaction, because it's not clear why for just to have some stupid filters, or excuse me, V1, you need all this local circuit, you need all this diversity of different cells, you need different layers. What is all this for if it's just to transmit a bank of filters? Okay. So let me now switch to network theories. Now, network theories are harder to classify, but I would like to, anyway, to put them in some order. So there are sort of minimal networks theories which augment the receptive field picture, the filter picture of a single cell, by something which is beyond, influenced beyond the preferred stimulus or the filter that this cell is responding to in the linear part. So there are feed-forward inhibition, there are divisive inhibition. So these are the minimal models, and they look like that. So you have some image, you have receptive field, the linear part, which is convolving with the image, and then you have things which are beyond this K, this filter, which is either subtractive, sort of maybe feed-forward inhibition, or divisive, which is here doing response normalization, and then you have, after that, you have again nonlinearity. and then you have response. So these are minimal models because they don't really involve the full network dynamics. It's more like taking into account some influences beyond, or nonlinearities beyond the linear filter. Okay, now, and then there is a range of models which take more seriously the network approach and try to model V1 and a single neuron in V1 in particular responses as an outcome of the dynamics of the network. And I'm putting them in this order because I think they are interesting to elaborate on. So one set of models, I would say competitive networks, which I'll talk about them later on, but I just say that one aspect once we are trying to understand that the network dynamics in V1, we have to make some assumption or to make some measurements or to look at measurements and extract from them an idea about which neurons are connected to which and how they are connected. Competitive networks are networks which are assume that the lateral connections in V1 is dominated by lateral inhibition, but moreover that neurons which have similar preferred orientation or similar preferred features are more inhibiting each other than others. And as I'll talk more about later on, this has shown to generate some sort of a winner-take-all or sparse representation of independent features. Okay, but it's interesting that it has this architecture which is orthogonal to what you might expect from the slogan "fire together, wire together," because here neurons which are firing together are actually inhibiting each other. Okay, so that's the competitive network models. Now, the ring network has a different assumptions about what is important in lateral cortical connectivity. It assumes that there is inhibition here, there is excitation. Again, the ring metaphor is because now we talk about orientation. We take orientation column, we put it on a node in a ring, and we just model the entire hypercolumn as a set of orientation columns which are interacting between each other. And because of the orientation symmetry, then it has the topology of a ring. But in any case, the assumptions about the connectivity is a Mexican hat connectivity. Namely, you have inhibition, which is maybe stronger for similar orientations for nearby columns, but you have excitation, which is also stronger for neurons which are at nearby columns, and the balance between them gives rise to a net interaction which is excitatory for nearby columns and inhibitory for distal columns. So this is the Mexican hat profile, which is quite different. Functionally it does different job than the competitive network that I mentioned before. It is really dominated by the recurrent amplification due to the cortical excitation, and which is sort of controlled and suppressed and stabilized by the inhibition. In any case, there has been several studies for such a network. The advantages are it amplifies signals, particularly it enhances selectivity, and as I said, it's unique among many models of V1 local circuitry in that it places important a role to the local recurrent excitation. Okay, now let me just mention briefly balanced networks. Again, has been a long tradition of models of this sort. The connectivity can be of this sort, but it is in terms of the functionality, it assumes that what's important is actually the feed-forward excitation. So you can think about this as actually coming from the feed-forward excitation and the feedback inhibition. So it is a balance between a very strong feed-forward drive to a neuron or to the network and a very strong feedback inhibition. And what's important here is the sparse and strong E&I connectivity. Now, in terms of single-cell responses, it has explained several things which are useful for V1, the linearization, sharpening of tuning, etc. I won't go into this, but I want to just mention that this type of rather simple networks, sparse, strong connectivity of neurons in a very simple architecture, unlike previous models, has the advantage that they sort of for the first time really make predictions not only of sort of static responses, but also of the network properties and the dynamics of that. Fast responses, temporal irregularity, trial-to-trial variability, weak correlation, so the state is asynchronous state, and also the origin of heterogeneity, why there is a diversity of neurons with different mean firing rates and different many other properties. So there is a whole range of properties of the ensemble of neurons, of the population of neurons in V1, which are explained by this very simple model. So it goes beyond explaining the orientation tuning on the contrast response or single cell properties. Okay, so what are the problems of this entire class of models? First of all, they have not been tested on natural stimuli. So one cannot really say whether such simplified models of network dynamics in V1 are adequate to explain responses to natural stimuli. And the reason is not because we have been lazy, it's because there are many parameters in such model, because you go beyond, in addition to all the parameters describing the feed-forward filters, you have now to describe the pattern of you have to assume into the network the pattern of connectivity. Although these models are still very simple models, still those many parameters are such that it sort of deters us from actually going to make a quantitative comparison with responses to natural stimuli. Other properties, other problems have to do with insufficient anatomical physiological validation. We still, today, I would say, I think it's fair to say, we don't really know whether local cortical dynamics is dominated by excitation or by inhibition or by balance between them or by both of them. And again, there are very prominent schools that basically advocate that what cortex is adding is inhibition, primarily inhibition, and that's one, I think, of the most outstanding questions, which is still missing, is a clear physiological validation of the role of excitation. Another question is the pattern of excitation versus inhibition. Is inhibition longer scale, as advocated by the Mexican hat model, that excitation is narrow, inhibition is broad? Are inhibitory neurons tuned or not tuned? So the whole question of the differential properties of excitation and inhibition, both in terms of connectivity and in terms of properties of neurons, are still not clear, and there are contradictions between different experiments or different labs. Now, another two obvious restrictions is one, that most of the work on network models have focused on local circuit. It's not exclusive. There are interesting extensions to several hypercolumns and to horizontal connections, but it's fair to say that most of what we understand a theorist about what would be the properties of a network dynamics of V1 is still restricted to the local circuit, and in terms of functionality, I would say mostly focused on single cell responses. So we have network, not all the models, but many models have writing down a network model for V1, but then they say, okay, what is the network predicts about the tuning properties of a single cell in the network, and compare it to the experiment. So the network is here as a mechanism of explaining the properties of single neurons, but still the focus is on the single neuron as a unit. And what is missing, and I think Bob already mentioned this, is a more explicit focus on pattern of connectivity, of activity as the units of computation. And again, because of maybe lack of idea or maybe lack of sufficient data, this has not been done in sufficiently enough clarity and seriousness that we can say something that we understand that. And if Tali mentioned that that students need to come up with a research proposal, so here's a question: why not asking about the receptive field of a network of V1 neurons? What is the receptive field of a network? I think if you think about it, the students that took Reshatot Neuronim Aleph and Bet and Gimel and so on, they should be able to come up with a research proposal proposing in an intelligent way to describe, to study, to investigate the receptive field of a network of V1 neurons. But this is kind of thing that we miss from the discussion, either experimental or theoretical. Okay, so now I want to switch gears to another style of models. These are models which I call normative models. I miss here a slide. Maybe I jumped. Okay, no. Okay, so I miss your slide, but I want to. It's important, so I'll just say it in words. What are these network models attempting to achieve? You remember when I asked about receptive fields, I said, well, this is what they try to achieve. And I think, and I want you to be aware of that, of the network model that I was describing so far. Basically, have this as primary goal, compact description of responses, but maybe at the network level. So the phenomenological aspect I want to emphasize. The network model that I was describing, and I think the most important type of network models that are relevant to understanding what V1 is good for, is sort of simplified or abstract, whatever you want to call it, phenomenological models, which will give us a powerful but yet very compact description of what the network is doing. Mechanistically, they are attempt to, as I said already, to explain how some aspect of the responses of neurons come from, not only feed forward but feedback and so on, and function that to some extent those networks suggest function for the circuit. For instance, I said cortical amplification, sharpening of tuning, maybe temporal irregularity is important for other things, and so on and so forth. So we can digest different models according to those axes, and I think that's important to bear in mind. Okay, so now let me switch to other type of models, which are models which are normative in the sense that they are not trying to describe what neurons in V1 do based on experimental data and connectivity, but they try actually to predict or to design V1, if you want. And they are asking the why question, not the what or how, but the why question. Why are V1 receptive fields are as observed, and so on and so forth, or maybe also why the connectivity is observed. So basically, they are attempting to give us an answer of the functional role of V1 as not as a single neuron, but as a whole, as an entire system, and to approach it from purely computational perspective. Okay, now there are several types of these models. Efficient coding theory, starting from all the ideas of Barlow, and then Adick and Redlich, and then more recent in the 90s, Olshausen and Feld and Bill and Sejnowski, and ongoing version of them, for instance, Yair Weiss and collaborators. they are basically telling us, I think most of them, that V1 is trying to generate sparse over complete representation of independent features or important features. Sparse means that neurons in V1 are claimed to be responsive to a more select set of stimuli than neurons in earlier stages in the visual system. And over complete is meaning that the number of neurons in V1 are substantially larger than the number of axons coming from AGN, maybe by a factor of 20 or 50 or 100, depending how you count them. So there is an expansion of the dimensionality. So the visual image is now represented by many more units or filters than the input, and each one of those is sparsely active. Okay, so here's an example, natural images. This is in independent component analysis. If you take natural images and you apply those computational efficient coding theories to actually ask what would be the best representation of natural images, you come up with generically with these Gabor filters as opposed to the PCA filters. This is well known now. So, and this is one of the great successes of the theory. It explains, so to speak, why the filters of neurons in V1, or let's say simple cells in V1, have these Gabor-like filters, oriented filters. And again, the idea is that those filters, those Gabor filters, represent, or the origin of these Gabor filters come from the fact that natural images have edges as the elementary or part of the elementary of the vocabulary, of the dictionary of building blocks, edges, and these are represented by V1. Now, of course, everybody from human vision know that, well, I mean, we have this receptive field to detect edges, but the success of this efficient coding theory is to show that you should start from natural images without positing edges. You just by blind demand of having an efficient information, let's say information theoretic efficiency or other criteria of efficiency of coding, you generate those filters. Okay, so now what do these theories tell us about the actual mechanism of generating those filters or those sparse representations? Now here, the picture is, I'm going back to this competitive network, and basically you can think about it in terms of this simple diagram or simple equation here. You have a neuron in V1 in this model. Its activity is given by this A1, the firing rate, let's say, A1, A2, etc. There's an image S of T, S is the image, and there is a bank of filters, phi 1 to phi m, which describes the feed-forward projections, the feed-forward receptive fields. And then you have this synaptic input, embedded potentials, U to UM, which have this simple dynamics, and then there is some nonlinearity to generate the firing rate. So what's the reason I mentioned this is because this model suggests that there is a relation between the feed-forward filter phi of each neuron in V1 and the lateral inhibition between neurons in V1, which is given by the phi transpose phi. So which means that neurons which have highly overlapping linear receptive field, feed-forward receptive fields, are more inhibiting each other. This is, if the dot product between the two neurons is large, then they are providing inhibition, and then there is a leak. Okay, so this is the basis of this competition, which means that fire together inhibit each other, fight against each other rather than wire together. Okay, so it is interesting because this computational model provides some clear prediction about lateral connections. Has this form, and it's a prediction that one can test in experiment. Okay, so efficient coding theory of this type. Now, this brings me to a more recent and a very sexy set of models, which are under those lines: generative models, predictive coding, Bayesian networks, etc., etc. And to motivate this this kind line of thought, let's go back to this dynamics. So if you think about this network, you can think about this as feed-forward input, as I said, and then there is a lateral inhibition from filters, from units where corresponding filters are highly overlapping, so the dot product is large, so that's fine. But you can rewrite the entire, the same network, you can rewrite dynamics, you can rewrite in the following. You can take these two terms and combine them, and then rewrite this network equation in the following way. U dot, the input, moment potential, is now E of t minus U. U is the leak, and E of t is this two term, and there is the linear filter, the feed-forward filter phi, but it's multiplied now by the image itself minus the contribution from the lateral inhibition, which is the different phi's for different neurons multiplied by their activity. So this is the feedback inhibition. And you can think about these terms as the net term as an error term, where this is the activation coming from the image. So this is S of t, and this is the predicted image, the predicted by the activation of the activity of the other units. So, that's interesting because it means that what is activating at any given time, so to speak, each one of the neurons is a predictive error, is not the actual input S of t which drives the network, which drives the neuron, but because of the feedback inhibition, it is the difference, the error between the actual image and the predicted image coming from the lateral activity. Okay, so this is why it's related to generative models, and what does it mean, generative models? So you can think about this process as representing or emulating the actual production or generation of the images themselves, because under such model, the images are actually produced by linear combination of basic elements like edges or other elements, maybe faces in higher order areas, times coefficients A, which represent the mixture of those elements in any particular image. So you are under this description, the brain tries to emulate the way that the images are generated. This is the way the brain predicts this image. So the sum of those filters times the activity of the neurons is the predicted image, because it emulates the statistical model that generates the image. And according to this picture, the business of the cortex is actually to compare the actual image with what is predicted, if the filters are, of course, are the right filters, through the activation of those A's. And the A's are called the causes, the independent causes of the image. Okay, so generative models, predictive coding, and Bayesian networks. Now, why Bayesian networks here? The reason where Bayes coming into the picture is because these causes are, at least under some models, are couched in statistical framework. So in the sense in which these causes are independent is in the sense of the generative statistical model of the images, And naturally, when you have such a picture, you need to make some inference about the statistics, and that's where, under these frameworks, Bayesian networks come into play. And that's, so I'm sure that Tali Tishby is very happy about predictive coding. I'm not going to talk about action, perception action cycle, because I'm not talking about action. But I would like to talk about extension of these models to hierarchical models. And I think that's sort of what is the state of the art of model of view 1 in the context of the entire hierarchy of the visual cortex. And it has been proposed by Ballard and company and Carl Friston and others. And basically the picture is, cascading what I just explained. So you have some input, the actual sensory input. This generates two signals. One is initially, so to speak, is the input itself, but at any given time, the feed-forward signal is basically the error signal, and at the same time there is a prediction of what should be the input, which from the higher level, from this level. So at any given time, you have from the inhibition, as I explained, you have a prediction what the input should be, and you have the actual input, and that generates the residual error. And then the next cascade, you have the same story. You have the signal, and then a prediction of what the signal should be, and so on and so forth. And this is sort of example of how this will work for several layers in a cascade. So you can imagine that edges are, for instance, are independent but conditioned on contours. And contours themselves are more complex and larger objects which are independent. So you have, in an image, you have independent causes on top of the layer, which are the activation of contours, and then you combine them to get complex cells, simple cells, edges, and then the image itself. And what is proposed in this model, which is, by the way, related to some extent to the reverse hierarchy theory of Hochstein and Achisar, and this is that there is a very strong role for the feedback, not only in context, in the sort of modulation and attention, but actually in the actual processing of an image, because the actual processing of image, let's say image recognition, is a combination between the feed-forward flows of errors and the feedback generation of predictions of the image. So that's a very nice story. It has extensions also to action, but I won't get into it. But I would just finish by saying that there are, despite the appeal of that theory, I think there are still many lacunas in this, and I think at this point is more speculative than seriously based on physiology and anatomy. For instance, under some descriptions, not all descriptions, it requires that there will be special error unit and representation unit. Again, depends on the model, but some models require that. There is no evidence of such description. There is, again, many of those models are based on probabilistic inference, so the brain has to do both to represent probabilities, but also to update them in sort of Bayesian way, and there is hardly good, reasonable suggestions of how neurons or circuits in the brain do such computations. And again, there is no anatomical physiological validation of that role of feedback in sensory processing. So I just want to mention briefly that when we talk about generative models, then it does take the view that the brain is always making predictions about what next will come out, and these predictions is coming partly from the immediate experience, but partly from memory, etc. There is another complementary view of the brain as a generative machine, and that's sort of more from purely or more conventional nonlinear dynamics approaches, where the cortical dynamics will naturally give rise to attractors or attractor manifolds, provided again that the excitation is the dominant factor, not inhibition, because when the inhibition dominates, it's hard to generate those attractors. And again, under the attractor picture, indeed, the brain has either active or at least potential already limited set of states available for processing, and those states are selected for by either sensory input or by top-down signals. So it shares some aspect of the previous models in that the brain has something to say about what is going on now, and it's not simply the activity is not simply generated by a set of propagation or filter activation, by the actual dynamics which represents what the brain is expecting, or the potential possibilities that the brain is expecting. Persistent activity already Bob mentions, and of course, if really turns out that the generic network cortical dynamics has this potentiality, then pattern recognition and completion and memory and link to memory is a natural consequence, as Bob already mentioned. But I would say again, there is no concrete proposal of how V1 will work together as a attractor or pattern recognizer together with sensory processing. Are they two different aspects of V1, or maybe it's not V1, etc.? These are all open questions. But it's important to, I think, to highlight the fact that there is a mechanistic or dynamic substrate for cortex to actually work as a generative machine, and you don't have to posit some hypothetical Bayesian units in the brain. Okay, so briefly then, detailed models that I didn't mention them because they have very different agenda in my view. They really want to reconstruct the cortex of V1, and there have been some interesting attempts to do large-scale modeling of V1, the NYU, Ken Miller and company, and many others. Idan Segev, Henry Markram, and again others are busy working on this. The problems here, I think that are well known. Many parameters are not known. Goals are unclear and relevant to function. I think that's a very serious problem to be understood or to be argued about this approach. But I focused my presentation on approaches which try to come up with a phenomenologically compact descriptions of neurons and circuits and tightly related to their function. Okay, so now what are the outstanding issues in my mind? On several occasions here in debates, I mentioned my serious concern about the state of theory or the state of research in general, not only theory but also experiment in system neuroscience, particularly in cortex and in V1 as an example. And this is that we don't really have good, solid evidence, experimental evidence for what are the relevant modules. So we have models and theories and so on, but is the column a unit, a functional or anatomical unit? Is a hypercolumn unit? Different layers? Are they separate units? And maybe groups of cells which have different, it's the same morphology or some neuronal clusters and so on. So these are, I think, a very important and outstanding problem. This is within V1, but similar questions about V1. Is it meaningful to ask what is V1 doing? Maybe V1 is not a module is not really a model by itself. It's part of a collection of networks which are connected to each other and talking to each other, and together they do what the brain needs to be do with the image. So cortical and subcortical feedback, as already Bob mentioned. Now more methodological questions, which are of course related in, is adequate level of description. When you take receptive field, then you add all kinds of gadgets and tweaks to it and tweaks to it, and then what are you trying to do? Are you still trying to capture some compact description, or you're trying to basically give a full table of description of what the neuron is doing? And similarly with networks. So what is the adequate level of description? Related to it is when we compare models or compare approaches, and sometimes we fight among ourselves about those. It's not clear whether they are really addressing the same question or they are talking about different questions. So for instance, the feedback. So you can maybe suggest models in which the feedback is actually important part of the rapid processing of an image, or you can say, well, feedback you can have another model which describes feedback in terms of context or attention and so on, and these different approaches may not be mutually exclusive, but simply asking different questions. And this is basically brings me to the question, which is overriding question: are we asking the right questions about V1? So now some of these problems are fundamental problems for theory, and I admit that we have to do, in my view, a better job in coming up with methodological questions. We have ideas about predictive coding and Bayesian inference and attractors and many great ideas, but I think there are underlying methodological questions which have to be addressed before we can put to test those models and theories. But I want to say about experiment. I think what is the experiment? I don't have suggestions for one experiment or two experiments. I think we have to really revolutionize our expectation from experiments. I mean, it used to be that you will read the paper and say, well, there are 10 points on the figure on the graph. I want the 11th point. Forget it. We're not going to make progress by doing the business of physiology and anatomy as in the olden days. We have to make progress. I believe we have to, in addition to the fundamental questions which I described, methodological, theoretical, we have to come up with answers. I mean, experimental answers. And experimental answers are not going to be from sparse measurements of some neurons which are unidentified, or few connections in millions of connections in a network. We have to be able to, for instance, reversibly isolate pathways and systems that once for good, we will be able to say, is feedback from V1 to thalamus important or not? Okay, so what is the experiment? Turn off the feedback in a behaving animal reversibly, not giving time to the system for a month to adapt and to rewire to auditory cortex, and then measure the consequence on the ongoing neural responses in V1 and downstream, and the behavioral consequences. There's a simple questions which we need simple answers. Now, of course, millions of dollars and many people and work and technological hurdles, but I think that's the kind of results that we need from experiment. Similarly, about the representation, I talked about research proposals for population or network receptive fields, but what would be the experimental verification or validation? We need to record dynamics of maybe not all the neurons, but a large fraction of the neurons in a local circuit, in the resolution of a single cell and single spike and single synapse, no less. Okay, and I think the technology today is coming of age, and I think it's maybe possible. And similarly for patterns of synaptic inputs, either intercellular recordings or by activating or by observing dendritic inputs, to actually come up with definite answers about at least the functional architecture of who is connected to who in the local circuit, and also about feedbacks as in point one. So I think we have to stop giving ourselves excuses. The questions are simple, and both experimentalists and theorists have to join together and to come up with answers to these questions. Thank you.
I actually have a long list of comments and questions for you.
Okay, very good.
But we don't have the
Well, we have
one small one. I mean,
okay.
there's no need to see error units in the brain in order to have things like log-likelihood ratios, which are very easy to see and a lot of people feel. But now I want to open the floor for discussion to both speakers. Maybe we start with both.
Well, I just wanted to respond to the dialogue aspect of this whole approach. There's one particular thing that Haim brought up that I wanted to address, and that is this issue of the relative strength of excitation and inhibition in V1. Because we do have some experimental results that speak to it, and I thought I just want to put them on the table and see what you say and what other people say about them too. And this has to do with experiments that Jun Jing did with us and Dario on measuring the dynamics of orientation selectivity in slightly anesthetized monkey, single cell recording, and by using the reverse correlation technique, you can look at the time development of orientation selectivity. So you get a kind of a time course, and you can see that in many cells, what happens is that as a function of theta, the orientation selectivity looks like this at some early time, and then at a later time, it looks like the whole curve just translated down. It's supposed to be just a translation. And then later, it'll look like this, where this is 0. So there's a kind of a uniform depression, a kind of a suppression that's uniform over all orientations. And then you see other cells where it looks different, where the early case may also look like this. This is early. This is not early vision, but early in time. Early time, peak, say, and late. And then in other cells, you might see things that look like this. And then, this is 0 again, and then it might be, So, these are more common than these in our sample, but you can see both types, where you can see a sort of Mexican hat in some cells, and this sort of more global inhibition in others. In general, in order to fit these kind of data, you can do a good job assuming that the excitation is already set up early, and that with time all it develops is inhibition. Inhibition that either has, so that the orientation dependence of the inhibition, now plotting it positively, so again, theta, in this case would just look flat, and in this case, the inhibition would be peaked.
What's the time scale?
This is the orientation dependence of the inhibition, and then it varies with time with some time scale, which you can also draw. I mean, so the time scale, if we draw it in time, I didn't do it. All of this is sort of snapshots. If you draw the time dependence, the excitation looks something like this, and the inhibition is a little bit delayed. The untuned inhibition, which I'm drawing lots of blood. The untuned inhibition is a little bit delayed.
The question was what is the time scale?
What is the time scale? Yeah. So, sorry. So the time scale, this would be like 50 milliseconds, roughly. And then the tuned stuff is actually a bit slower, like that.
But isn't it surprising that inhibition is slower, unless the excitation is peak forward?
Well, what you can the observable is that at early times or I should say as a function of time, the response at the preferred orientation and at the orthogonal to preferred orientation are basically on top of each other, and then with time the non-preferred orientation response goes downward and departs from the preferred. So this is the idea that there is some kind of fast early excitation, whether it's whether the excitation could include some recurrent excitation that's fast also, but it's very broad. And then there is this broad inhibition which comes in with some delay. In general, we can fit most things just with this sort of somewhat delayed inhibition. So that just speaks to this issue about the inhibition. That was the main thing I wanted to bring up. And then, there's one other thing. Sorry. In cells like this, it turns out that you could divide the inhibitory component into two components, one untuned component and a separate tuned component. That is to say, they were both, the curve fitting could be accomplished with two components. With these different time courses. That's how you could separate it. And then you could do experiments, and what the experiments were done in this paper, and this is one of the nice things about the visual cortex compared to, say, other systems, is that you can change the amount of input by changing the size of the stimulus. So in one case, you could have, you could use just a small stimulus, which was something like, half a degree. And then you could do another experiment where you have, I think it was something like 3 degrees or something like that. And you could do the same experiment with both different sizes and see if there's any change. And what you find is with the 0.5 degree stimulus, you always get this pattern, and it doesn't change actually. And for these sorts of cells, it doesn't change when you go to 3 degrees. But for these kind of cells, you also get this pattern when you use 0.5 degrees, and when you go to 3 degrees, you get the Mexican hat. So our conclusion from that was that the what we call untuned suppression, or the global inhibition, whatever you want to call it, is actually confined spatially to just the optimal receptive field, the classical receptive field, and that the tuned stuff is actually coming from the longer distance in cortex, or maybe by feedback. So by using the spatial aspect of the stimulus, you could dissociate the untuned and the tuned suppression elements. So our conclusions from all of these ideas, all of these experiments were, and it does speak. The reason I bring it up is just that it speaks to some of the major issues that Haim brought up. Is that there is a lot of inhibition, and it seems to be generated in a local circuit manner, and it seems to be fairly broad. But there is, in fact, a tuned or a competitive inhibition, or what we call it, it's something that will generate a Mexican hat, but that doesn't seem to be in the local circuit. That seems to be coming from long distance, either from horizontal connections or from feedback from MT or from V4 or something like that. And so I'll stop there. But just these are comments that he sort of evoked from me. I couldn't stop.
All right, thank you. So let me open the ground for some other questions.
I'd like to comment about this is a question for Haim, relating to the last slide. What should experimentalists do? So you made one point that you made is that in order to study the connections from the V1 to the thalamus, you need to inactivate these connections reversibly to see what happens. And my comment is, well, you don't need a theoretician in order to make this comment. Second is to measure.
What is the question?
Let me make the second comment and then ask the question. Second point was, well, you need to measure, this is for the experimentalists, you need to measure for more neurons, better temporal spatial resolution, and so on in the awake behaving animal. This is again not something that you really needed a theoretician in order to make this inference that it would be better to have better spatial temporal resolution. My question is, what are the specific contributions of theory to the experiment that need to be done?
To bang on their head. I mean, but that's my point. My point is, every theory has some specific predictions. But so what? I mean, how can we make progress? So I have a prediction about the current excitation, and then you heard the story. Okay, so is it an answer? Well, this is layer 4. This is well, there is an excitation, but you don't know if it's horizontal. You don't know if the early excitation is feed-forward or incoming. I have a very specific question coming from the model. These answers are not satisfactory, unfortunately, because they don't don't have the capability, or they have the relatively weak capability of dissecting the components of the synaptic action. They have to some degree, but only to partial degree. I mean, even here you don't know whether there is a long range or feedback or something else, and so on. So every theory will have specific predictions, okay? But I don't think, I don't find the entire situation satisfactory unless there is a capability of getting answers to this. And my point, this is my point. My point is that most theories about cortex make predictions about who is connected to whom. Okay, is it mostly inhibition, is it mostly excitation, so on. Some theories say something about feedback. Some theories say something about timing, and so on. So I can, every theory will give you this, but that's what we have been doing for 20 or 30 years. But the point is that the answers are either not forthcoming or are coming in a way which is partial, and in a way which is debatable, and in a way which different labs contradict each other. So are we going to just continue doing it? I don't know. I find that the most important thing is to highlight and to the fact that we have to really change the way experiments are actually acquired, or actually give answers to specific, very fundamental architectural and functional questions about the system. And then we can argue, whether these responses are error or not error, so whether log likelihood, or whether you need to sample probabilities or not. Each one of the theories gives some specific predictions. And I'm not committed to particular theories. I'm not giving you my particular list of things. I think the most important issue is not to have a better, to have completely different. I think in this list, I'm not talking about having better results. I'm talking about giving complete answers. So we have to be in the neuroscience in a situation where there is a question which is interestingly enough, and one experimental lab claims to have the answer. First of all, other labs try to reproduce it. And secondly, the outcome is a V, and not a question mark which will go on and do something else. And that's the only way I can see it's some change of mind in the field, but also I hope that with the new technology, this will be, answers like that will be able to come up. Okay.
Somebody else?
I'm sort of disappointed by the interaction. I would expect you to say that only if we have a good theory will we know what experiment to do. Not that we have to do experiments which are more detailed, and we have to know more facts, and we have to record from many cells at the same time and know all the details about them, but rather only if we have good theory can we do the right experiment to find out if that theory works.
We have good theory.
So then give the specific experiment that has to be
I
done.
A specific experiment is what is the rules of connections in V1, local connections in V1. How are we going to do the answer to that?
The answer to that is what connection do you expect, and we'll test to find out if that connection exists or doesn't exist.
How do you test it? In monkeys, you will not be able to test it. In cats, you will not be able to test it. That's not. You're able to test it only in species and with technologies that will be able to map those connections. That's my view. I mean, there are a beautiful theory. I'm not connected anymore. A beautiful theory which have made very, very specific predictions. I think the answers are always dubious. I mean, this is why with orientation selectivity we're still debating about it. What is the role of cortex? What is the role of recurrent excitation for orientation selectivity? What is the role of inhibition? I mean, some claim that it's completely feed-forward to this day. But there are theories. But the point is the experimental data is so sparse It gives rise to multiple interpretations. That's my point.
But maybe it is the problem of the theory. Again, I mean, you listed here at least 3 or 4 different theoretical approaches.
Right.
And they can't be discriminated on the basis of the current experiment.
Well,
some of them are simply not being tried.
As I said, okay, let me just say, as I said, not all of them are mutually exclusive. So it's not fair. There are different models, there are different models, but some of them are contradicting each other. But some of them exclude each other, but not all of them. So, for instance, if I talk about the recent, if I talk about, in my view, the generative models, I don't think they are sufficiently defined or refined in such a way to make predictions about the local cortical points. But the more basic models, starting like sparse coding models, make very, so you have 2 theories which make very strong and contradicting predictions about cortical cells. Okay, so there are predictions, and there are, I think, good theories. Not infinite number of them, but some candidate theories, which to some degrees give orthogonal or conflicting predictions. But the question is, how do we go about answering this? This is just an example.
So I don't understand why, measurements that show that, 90% of or 80% of the neurons don't have this sort of orientation selectivity for the inhibitory components, why that doesn't rule out this kind of theory. I have to say, it escapes me. Because it seems to me, you can criticize rightly, I think, that we can't say very strongly what the recurrent strength is, although I think we could argue about that. But about the inhibition, there's a lot of, very broadly tuned inhibition.
But
in terms of orientation.
But there are also Shapley tunes.
No, well, there are some, but I'm saying, but the generic curve doesn't look like that. So why doesn't that rule out, that result rule out this theory?
Well, if I remember correctly, experimental data, intercellular recordings. Let me go back. Intercellular okay, let me just be here. Where is it? Yeah, where I was. Here. If you look at intercellular recordings from some labs, and I think even Förster will say that the inhibitory part of the synaptic input is tuned as excitatory, at the same.
Well,
we can spend now entire evening debating whether we believe Förster results or not. That's the point. And Förster will say, I don't believe Bob Shapley's results. And somebody else will say, well, or both of them are anesthetized animals, and I don't believe any of those.
Yeah.
Okay, so what should two theories say?
But the thing is, you can find, certainly in anesthetized animals, you can find orientation selectivity. You can at least try to, explain orientation selectivity under the conditions of the experiment. I would say that, actually looking at Förster's data and looking at other people's data, there's a lot of consistency, that there's a lot of stuff that's not consistent with that picture.
All right, one more question.
First of all, I don't know what's the difference between a theory and a model. And secondly, are these theories and models purely descriptive? Do they have, is there a way of distinguishing among the different models, the different theories, experimentally? Or is it simply a matter of measuring the parameters of the universal model?
Well, I'm not sure okay, so the difference between theory and model, I would say theory is more, in my choice of terms, theory is more a set of principles, and concrete implementation of it in a mathematical set of equations that you can either analyze or simulate. That's what I will. That's the way I use the terms, but I don't want to. What?
Basically, we agree on that. Theory should be a higher level and much more compact set of.
So in this picture, the idea that the view one of the brain is doing I don't know, a representation which maximizes some information theoretic measure subject to constraints, I would call it a theory. But an implementation like this, I would call it a model, because it's already a concrete implementation, some neuron-like unit. But again, it's yes, okay. But again, but about the second question, there isn't only one way of testing a model or a theory. But I don't want to distinguish between them for the purpose of this discussion. Obviously, a model can make different predictions about the behavior of the system or also the behavior of the animal. According to some models, if you show I don't know, competing signals, the brain will do this, and other models will say the brain will do something else. So obviously, this will be, in some sense, the best case, the best test of models where they predict something about the behavior of the system or the behavior of the animal under certain conditions, and that's sort of maybe the easiest way to test them. But many models I don't think are ready for such tests, because what they actually use as the inputs is already some well-cooked set of knowledge and data about neuronal representations, neuronal responses, and which with themselves have been tied to some degree with behavior. So under these conditions, or under these situations, the models make some assumptions, and you can, if the assumptions are essential enough, you should test the assumptions. I mean, that's another way of testing it. So there is no one way of testing models.
Do these models differ fundamentally or parametrically?
We don't have a standard model of V1, which would then just look at parameters. I think some models are fundamental. Well, I don't know. If the question is how fundamental you want it to be. All the models are neurons and have synapses.
I think that the fundamental difference in a top-down and a bottom-up model. I mean, in the sense of
so
something like this.
So that's an example. Okay, so if you think that actual to recognize from an image, to recognize whether the image has an animal in it or not, fast object recognition task. According to some description, this is purely feed-forward, and the feedback that is not involved in that. Okay. Other models presumably will rely on feed-forward and feedback even under these conditions, because they are the essential part of what's going on. So this will be fundamental, but a very important distinction between models. And what is the test? You know, simply abolish the feedback and see what happens.
I think actually this is an important question to explore, because I always feel that when you come from biology, really this distinction, what is the role of theory, what is theory at all, or what theory has to give biologists is not really clear, and this is one of the main goals of this type of discussion. So I think this is an important question, and for sure it comes up so late. Anyway, this Bob has another talk to give in half an hour, so I think we'll stop here, and thank everybody.
Good.
Well, it was fun.
Yes, it was. A little excitement.
Good question. Good question.
Hi. So we're one third of the way through. Hello, can you hear me? Hello? Hello, what, can you hear me now? Okay. We're one third of the way through. Okay. Can you hear me now? Okay. So the thing is, we're going to go to dinner at 7. And we'll pick you up at the front door at like 6:45. The below. I'll come and get you. Either he'll drive down or I'll come down and get you. We'll walk up to the car. Yes. Can you hear me? Lori? Sorry, can you hear me now? Okay, now let me start again. I touched the phone. I don't know how to do the phone. Right. Just by the desk downstairs. Right. No, maybe like 10 to 7. Okay, and then I'll come down and get you, or he'll drive down, and then we'll go to the place which is nearby. Okay. Fine. We just finished the dialogue, and now I have to get prepared to give my hour lecture. But so far, so good. Yes, very much. Yes. No, absolutely. That's why we're definitely going to aim for something strong tonight. But so far, so good. How is your day so far? Oh, good. Well, actually, I did get a nice view of the city from Scopus, which was part of the visit this morning. So that was good. Okay, so I'll see you in a little while. I have to get my stuff ready. And hope you're okay. Take it easy on the knee now.